I welcome you all, dear learners, with another session. And this session is going to be kind of a case-based learning, which is going to be from your general physiology related something to your cell membrane and transport across cell membrane. So I welcome you to Dr. Reena's Physiology, Conceptual and Integrated. Please log on to this Telegram channel or the Telegram group wherein you are provided with the notes which I take on this class. When transport across cell membrane, this itself becomes your LAQ. Draw a well-labeled diagram or describe is an important SAQ. You get SAQ on glutes, on simple diffusion. And I'm going to also at the end of this make you write 2 BAQs that will be on differences between active and passive transport and difference between simple diffusion and facilitated diffusion. So let's begin up with the case. There is a 43-year-old man who presents to the physician with complaints of epigastric pain. And after a thorough workup, the patient is diagnosed with peptic ulcer. He is started on medication that inhibits the proton pump of the stomach. So in this question, you are given the clinical picture, but you've also given the diagnosis. Because I am directly coming to what I'm going to ask or what I'm interested in asking to you. What is the proton pump that is referred to here? What type of cell membrane transport will be inhibited when you are using this medication? And what are the other types of transport mechanism across the cell? Now you can see that there is a proton pump inhibitor, and this is the case of a peptic ulcer. That means if I have to put the same case in GIT, I will not give you the diagnosis that it is peptic ulcer. So अगर ये GIT में LAQ आना है case based, so I would ask more about the secretion of hydrogen. That is more important in this. But when I am putting you the case for a general physiology, I am more important in the various transport mechanisms. So to summarize, the disease the man is having is peptic ulcer, and therefore he is described a proton pump inhibitor, right? This proton pump inhibitor is going to inhibit the primary hydrogen ATPase pump. If you remember, the parietal cells, on the luminal border of the parietal cells, there is a primary hydrogen ATPase pump, which is very important for the secretion of hydrogen ions by an active mechanism. So you are going to inhibit this pump, which is an example of a primary active transport. So this pump is primary active transport. So please don't think that only sodium potassium pump is a primary transport. There is hydrogen pump, there is calcium pump with a primary active transports. Now, what are the other type of transport mechanisms? This we will bit elaborate, right? So simple, facilitated diffusion and so on. So यहाँ पर what was asked was basically about the hydrogen ATPase pump, which when inhibited by proton pump inhibitors, if you remember omase, tablet omase, omeprazole, tablet pantop, pantoprazole, these tablets, even we are known to these topics, and their primary mechanism is to inhibit the hydrogen ATPase pump. Now, why do you require this transport mechanisms? If you look into the cell membrane, it is semi-permeable, right? This is the hydrophobic part, and this is the hydrophilic part. The pin-like is the hydrophilic part. That means inside these are the fatty acid tails. Okay, these are the fatty acid tails, and they are aligned towards each other. So this structure of the cell membrane, which is a lipid bilayer cell membrane, is freely permeable only to fat soluble substances or to gases. So other substances, whether they are ions, whether it is glucose or any other solutes, they cannot pass through the cell membrane very easily. So for those substances which are water soluble, okay, for those substances which are water soluble, which cannot easily pass through the cell membrane, you require the presence of various pores, channels, carriers. and that is why we require transport across the cell membrane. This is the basis of transport across the cell membrane, that fat soluble substances can easily pass, right? But other substances which are there, which are fat insoluble or which are water soluble, they require carrier transports. So there are different diffusions depending upon whether the substance is fat soluble or whether it is fat soluble, that is lipid soluble, or water soluble. So you can classify the transport mechanisms basically into two types: active transport and passive transport. This is a basic classification which every first prof student must know. Your active transport is primary active transport and secondary active transport. The secondary active transport is either co-transport, where the two molecules are moving inside in the same direction, and therefore it is also called as symport. Then it can also be counter transport. Counter transport, where one molecule is moving inside and the other is moving outside. That means the two are moving in opposite directions, which is also called as antiport. So primary active transport, secondary active transport. Passive transports are diffusions and osmosis. Diffusion can be either simple diffusion, facilitated diffusion, or osmosis is the third one, which is especially for solvent. The other terms, for example, filtration, which takes place across the capillaries; bulk flow, where the substances are moving and the water also moves; for example, solvent drag. These are also coming into the category of passive transport. So passive transport में UG student के लिए simple, facilitated you must know. If you are able to remember filtration, bulk flow, solvent, it's well and good. But at least diffusion and osmosis, this should be known to you, right? Other types of transports include your vesicular transport, where you form a vesicle which is either thrown out or whether it is taken in, which is exo and endocytosis, and the transport across the epithelial cells or the epithelium, that is transcellular or paracellular. So when the substance is passing through this tight junctions, so this is one cell, this is another cell, you know that they are joined together by tight junctions. So when the substance is passing through these tight junctions, it becomes a paracellular transport. And when the substance is passing through the cell, through the cell, it becomes transcellular. So ideally, this much, or this is the most apt classification of transport across cell membrane. So A and B is must known competency. C and D you may know, okay, but at least even these vesicular transports are important. So whenever you are writing an answer on transport across cell membrane, please make sure that you cover all these points, because it will never be to describe them. It is just a classification and you are always asked to describe any one. Now, as I told you, the very important 2 marks BAQ, which is this one, that is difference between active and passive transport. So active transport is active because it is using energy in the form of ATP. Passive transport is passive because there is no use of energy. Since it is using energy, it is a metabolic process, whereas passive is a physical process. Active transport, it is uphill. Why uphill? Because it is moving against the concentration gradient. The substance which is transported is against the concentration gradient, right? Passive, it is along the concentration gradient, from low to high, against the concentration gradient. Active can be further primary active and secondary active. Passive is diffusion and osmosis. So if you are given a 2 marks BAQ, any 4 points are suffice. If you want to make it more beautiful, you can write examples. For example, you can write over here the sodium potassium pump, right? You can write SGLT, sodium glucose co-transport. Simple diffusion, for example, gases, they show simple diffusion. Glucose, galactose via GLUTs, they show facilitated diffusion. The second very important difference which should be known to you is the difference between simple diffusion and facilitated diffusion. This is another very important one. Now both of them are passive, right? Both of them are passive and they do not require energy. Both of them are along the concentration gradient, but still there are differences. In simple diffusion, the rate of diffusion is directly proportional to the concentration gradient across the membrane and the permeability of the membrane to the solute. So it is just going to be from low to high, and it will depend upon how much permeable the substance is. For example, you know that gases, simple diffusion. Facilitated, here the rate of diffusion is governed by the kinetics of the carrier mediated transport. You know that the simple diffusion there is no need of a carrier because the substance that is transported is lipid soluble. Facilitated the substance that is transported is not fat soluble, so you require a carrier, right? And if the carrier is less, suppose there are 50 carrier and the molecule to be transported is 70, then what will happen? It will show Vmax. That means the facilitated is subjected to saturation kinetics. This is what is saturation kinetics. When the substance to be filtered or when the substance to be transported becomes more than the carrier protein. This is the reason that why glucose appears in urine, because in a patient with diabetes, the amount of glucose that is to be passed or that is to be reabsorbed becomes more than the carrier protein available. So this results in the failure of 100% reabsorption of glucose, and glucose appears in urine. So facilitated diffusion is subjected to saturation kinetics, right? Another important thing is it is also subjected to competitive inhibition. What do you mean by competitive inhibition? Whereas simple diffusion does not show, there is no competitive inhibition in your simple diffusion. There can be a carrier protein which can bind to glucose as well as galactose. So two substances or two molecules can bind to same carrier protein. That is one important difference between facilitated and simple. Another important thing, remember that although the facilitated is requiring a carrier protein, it is faster than the simple. So facilitated diffusion is faster than the simple diffusion. So though both of them are passive, they do not require energy, but they bear important differences. Example, gases or any lipid soluble substance. For example, even if you consider hormones like cortisol, it will show a simple diffusion, isn't it? Because it is fat soluble, right? But ADH, a protein hormone, will not show simple diffusion. These come as MCQ also. Facilitated, glucose and galactose via the glutes, and there are urea transporters which also show facilitated diffusion. So we've seen the transport differences between active passive, simple diffusion, facilitated diffusion, and another very important is difference between facilitated diffusion and primary active transport. Here what is the similarity? The similarity is both of them use carrier protein. This is the similarity, that carrier protein is needed. But what happens that even though the carrier protein is needed, the facilitated is passive, there is no need of energy. Whereas primary active transport is ATP dependent, it is going to cause a direct hydrolysis of ATP, a direct hydrolysis of ATP. This primary active is always unidirectional, whereas facilitated, it can be bidirectional depending upon the concentration gradient, which side it is more, right? So even this difference is also very important. So for a 2 marks BAQ, we write 1 point and 2 points with one example, and this gives you 2 on 2. So nowadays, since there is a concept of 2 marks BAQ and many of the times you get differences, it is very important that you must know these differences, right? So we've covered active passive, simple diffusion, facilitated diffusion, and facilitated diffusion with primary active transport. So for the next class, where I'm going to take active transport in detail, just begin with an MCQ. The sodium potassium pump ka what is the coupling ratio? 3:2, that is 3 sodium. You say it like this also, 3:2, that is 3 sodium outside and 2 potassium inside. 3:2, but 3 sodium inside and 2 potassium outside. 2:3, that is 2 sodium outside, 3 inside, or 2:3 but 2 sodium inside and 3 potassium outside. So the coupling ratio is 3:2, where 3 sodium ions are taking outside and 1 potassium ion is taking inside. GLUT is an example of this. Is a very important MCQ, and I tell you that even this glucose transport is very, very important for your NEET PG questions also. So it is an example of simple facilitated diffusion, transcytosis, or secondary coactive. The commonest mistake which is done is between A and C, because a carrier is there. So many of the times we are confused GLUT as secondary coactive transport. So remember, secondary coactive transport is SGLT and facilitated diffusion is GLUT. So this is all for the day. We meet on very soon in the next class, where I'll be taking active transport in detail. Let me know that you are liking, and let me know the topics which you want me to teach. Thank you, and let's make physiology easy and integrated, conceptual.